Welcome to the second annual Marvin Karasek Memorial Lecture. I want to welcome the friends, supporters, faculty, alumni, students, and other members of the Stanford Dermatology community to this second annual lecture. This is a really special lecture for us, and we have a wonderful speaker. We'll have some more opportunity to discuss some of the great work going on in the department. But I want to start by orienting you to the department and showing you a picture of the faculty from last fall's retreat. The faculty in this department are super excited about the work going on at Stanford, and they have now become the largest faculty in dermatology of any department in the world. And actually, that's the first department at Stanford Medicine to become the largest department in any field. And because of the way growth is controlled here, it may be the last department to become the biggest of its kind. Now, this is a pretty happy group, aside from Dr. Vasquez, who you can see somehow has started to squint a little bit during the picture. And I think this group is really happy because they're embedded in the heart of the Stanford Research Biomedical Enterprise. And you can see here where we are here on the main campus of Stanford School of Medicine. And one of the key programs at Stanford that brings together investigators from all the major disciplines in conventional biomedicine is the Stanford program in epithelial biology. It's grown to over 76 faculty from 26 basic and clinical departments. It's very fitting that I tell you about this because our speaker tonight is the co-founder of this program in 1999. A long time ago now. He looks a lot younger than that. He was in grade school when he did this. But this is a truly amazing program. And so one of the things that makes Stanford Dermatology special, one of the things that's helped it grow so big, is because it is leveraging the strengths of Stanford. If you wanted to have a key to open and unlock the door of all the magical and incredible things happening here, this program in dermatology has figured out how to do that. So those aren't just platitudes. Those can be supported quantitatively. And this is a slide that we only show in the family. We don't distribute this outside, although some of our peers have gotten a hold of it, to their immense jealousy and discomfort. So our department is not just the biggest, but in terms of publishing groundbreaking research, which you can actually quantitate that by the Institute for Scientific Information's ultra high impact journal metrics. Here are the top 10 peers combined, and here's Stanford Dermatology. And these are just the papers from the last year. And in fact, an ultra high impact publication has rolled out of this department on average every 36 days. Now, to put that into perspective, that's more than any department at Stanford and is truly amazing. And Stanford Bioscience leads the world. So this is an incredible group. You're seeing some of the translational work here at this discussion and poster session, and Dr. Oro is going to tell you the full spectrum of basic to translational advances. So in addition to publishing amazing scholarship, this department trains more leaders in the field than any other. Here are just the PhD students that are trained in the department. The department trains a very large number, many of which are here today. Maybe the PhD students can just, some of them can raise their hands or smile. Katie, there's Katie. Maybe they're just over at the food. That's where they tend to congregate. Anyway, it's amazing. And so the average of our peers is about 10%, the number that are trained here. So in addition to creating the best science and creating the best leaders, this department has the best supporting community of philanthropists, of donors, of friends of the department, none more so than the individual who actually endowed this lectureship, Marvin Karasek. Marvin was a true believer. You know, if you are working at a place and then you leave your life savings to that same place, when you know the bumps and warts, you know the difficulties, you know that that's a person who really believed. And that's what Marv did. He was a faculty member here beginning in the 60s. He made seminal discoveries in keratinocyte cell biology. and Marvin chose to leave his entire estate to create this lecture today. Now, Marvin knew exactly how much money we had. Al is smiling over there, our former chair, my former boss who preceded me in this role. Marv knew everything, and he still said this is the thing he believed in the most, that where he wanted his resources to go, because he knew it would transform human health, would help alleviate suffering, and would help make a difference to people. Now, my cousins, every time they come out to visit from the Midwest, they see all these shiny buildings and they see all this amazing stuff going on at Stanford, and they say to me, Paul, why do you guys need more money? You guys are loaded. Stanford is beautiful. You can't possibly need more money. And Jim Hobart over there has heard me say this recently, as did several other of our friends. I'm looking around for someone that heard this. My answer to that is, what Stanford is like a beautiful, beautiful garden. Stanford gives you the land to till. But Stanford actually doesn't give you the seeds. It doesn't give you the water. It doesn't even give you the fencing to keep the varmints out from chewing up your vegetables. Stanford is like this unlocked potential. And what our donors do is they make it possible to grow amazing things there. The soil is our amazing students and postdocs, our incredibly talented faculty, our proximity to Silicon Valley, and all the technological advances are being in the heart of it. But really, it's what our supporters do. And many of you here are incredible supporters, and that means the world to us. And I just want you to know that nothing happens, none of the trainees, none of the great advances, nothing we do happens without your support. So it really means everything to us. Now, I'm really privileged to introduce the Karasek lecturer this year, Dr. Anthony, otherwise known as Tony, Oro. Tony is the Eugene and Gloria Bauer Professor of Dermatology, and Eugene and Gloria Bauer are sitting right here in the front row. Could you guys just wave? So Eugene Bauer and his wife Gloria, who go by Jean and Gogo respectively, have a long history with this department. Like Marv, they were actually in this department. Jean was the chairman before Al Lane took the helm. And so again, when you have people that know the bumps and warts that are giving everything to support you, it's really a vote of confidence that there's just amazing stuff going on here. So Jean and Gloria endowed this professorship, and Tony is the inaugural holder of the Bauer Chair. Tony is also Associate Director for the Center for Definitive and Curative Medicine. He's co-director and the major force of the Maternal and Child Research Institute, which are helping bring advances that we get out of adult studies and model studies to support the well-being of children and to prevent them ever getting some of the diseases we work on. Tony has had more, I'm really making him squirm. I've known this man since he was a postdoc, so it's really fun to introduce him. It's really great to see him kind of wiggling around. This is great. Tony has really done some pretty amazing stuff in his life. He's achieved more in his young life than the vast majority of people in biomedicine will ever achieve. And he actually is up for a couple more new big awards, which I won't talk about, that I'm quite sure he's going to get this year. He's run the Sulzberger Award, which is one of the highest honors in the American Academy of Dermatology can confer. He's won the Montagna Award, which is also the highest honor I think the Society for Investigative Dermatology can bestow. He serves on innumerable boards. He's been on the study sections that really hand out the NIH funding, the National Institutes of Health funding. He's done truly amazing things. But Tony has been more than just a very successful person in his own right. Tony has been committed to making a difference in the health of his patients. He's a practicing physician, but also in the patients beyond which he could ever reach. The children and adults who suffer from an array of different conditions, including cancer and genetic disease. So without taking up any more of his airtime, it's my great pleasure to introduce Dr. Anthony Oro as the Karasek lecturer.
Wow. Thank you, Paul. After that introduction, I'm not going to say anything because I can't really add to it. So, but thanks very much. Actually, it's wonderful, and thank you for being here today. I'll try to talk quickly because the Warriors game is coming on. I know you want to talk, but it's actually an honor to be the Marvin Karasek lecturer this year. When I started my lab in 1999, 98, it was actually adjacent to Marv's lab, and his lab was there for 37 years as he was winding down his career. He provided all sorts of advice on how to start a lab, how to take care of advisees, and also great advice on biking, skiing, and how to live a balanced life, something I took to heart. So it was wonderful to have him as a mentor. So this is really appropriate for tonight. The other thing was that he was very passionate about being curious and instilling that in our trainees. And so, which is why I want to talk tonight about basically therapeutic strategies in medicine and regenerative medicine. Because we have a new revolution in therapies. And that is that if you have a disease where you can basically cut it out, for example, a gallbladder that you don't need, that's surgical. If you have a skin cancer, you can cut it out. If you have an infection, you can give it a drug, like a pill. And then the antibiotic goes away, the infection goes away, and you're good. Now, what happens if you need the organ that you're cutting out, and you can't give a drug because it's there for, you need it? What do? And this is in particular things like, for example, high blood pressure, where all of the pharmaceuticals say, oh, we're treating it, but you're taking that medicine for a long time. And so by the time you're 80, you're going to be basically taking an average of 22 prescriptions at a time. That's not definitive medicine. That's just a Band-Aid for the therapy. And so what we need is really more definitive medicine where one dose is a cure. In Duke basketball language, it's a one and done. We call it definitive medicine. And that's really what the goal is because there's over 10,000 incurable monogenic disorders, such as these are kids that we can diagnose them genetically. We can sequence them, we know exactly what the disease is. We basically say, thank you very much, we send them on their way, and they're basically a lifetime of palliative medicine where we can't cure them. All sorts of these different diseases, immunodeficiencies, blood disorders, we can diagnose them, there's 10,000 of them, and at birth, what if we could actually take out the stem cells, correct them, put them back, and save them from a lifetime of palliation? What if we could do that? What about common diseases with genetic components? One of my favorite, male pattern baldness. Your hair is going away. There's no therapy for that, believe me. What about Parkinson's? What about diabetes, where we can give insulin, but are we actually treating it? What if you could replace the beta cells of the pancreas with cell therapy and cure it definitively? And finally, heart disease. For those runners in the group, if your knee joint or back going out and you still want to run a marathon, what do? If you're a hockey player and you lose your tooth, you have one shot and then basically it's gone. So these are all, or burn therapy, right? So what can we do? We needed a therapy where we actually can take the cells out, manufacture them, or else take them, cure them, put them back in. This is called regenerative medicine. We know that we can actually do this because this is an axolotl, an amphibian, where if you cut its hand off, it'll actually induce a set of cells that actually have the ability to make any and all of the tissues in the body. It'll do it while you're watching and regenerate the limb as you watch it. Okay? This is called blastema. And these cells here, these pluripotent cells, have the ability, in the right context, with the right, we'll call them ingredients, they'll actually reinduce this limb and make it just like new. So why can't we do this in us like the axolotl does it? And so if you know how to do that, that's the goal of this regenerative medicine therapy we'll talk about tonight. Okay, and this actually it's possible. In fact, I'll show you how we're doing it here at Stanford. But this next generation stem cell therapy is made possible by 3 major technological advances in the field of biomedical research. And actually just so happened that all 3 of them were developed here, are not just developed, but actually enhanced and are here at Stanford at this time in sort of biomedical history. And because of these 3 technologies that we actually can now have regenerative medicine cell-based therapies made possible. And so that is the ability to understand the stem cell biology, that axolotl limb, understanding what those cells are that then reinduce. We'll call them pluripotent cells. The ability to actually edit the DNA, using a technology called CRISPR-Cas9, which we'll talk about, that actually can specifically edit and correct any genetic defect without leaving a scar in the genome. And then tissue manufacturing technology, where actually then you can manufacture cells at scale to then put them back in people. And so all three of these technologies are now available. So the problem is that they've been available in parts, bits, and pieces, and it's sort of like when you know a little bit too much to be dangerous. That's been the problem at other places in terms of stem cell technologies, because as you've heard about in the news, many people have been trying stem cells injecting. We have stem cells. They're advertising stem cell biology. We can inject your stem cells and correct your vision. But what happens is that if you don't have the right manufacturing technology, there's all sorts of injection clinics now, for-profit clinics here, Merchants of Hope in the New York Times, where people are saying we have STEM technology and we can help you, but it's not approved, it's not biologic, and so we need some sort of a center where we can provide reliable, reproducible, and manufactured drugs for our patients. And that is what we decided and developed here at Stanford, called the Center for Definitive and Curative Medicine. It really is, it's one of the first centers that's both the adult hospital, the pediatric hospital, and the School of Medicine coming together. We take discovery research in these three areas I mentioned, with all sorts of the different departments, including dermatology, and we develop a manufacturing facility here on campus over at California Avenue to develop reliable and curative therapies for our patients. And this is really one of the big initiatives that also in Congress to develop reliable and improved stem cell technology therapies for patients. And so I'll just tell you a little about that in the next few minutes. In terms of tissue manufacturing, we've invested in a good manufacturing facility on the edge of campus, where actually we can develop novel cell-based therapies where we can actually at scale. You know, it's one thing to do it on a mouse, but actually if you're trying to do this where actually you're manufacturing massive quantities to squirt on patients, that's a whole different issue. And so it really requires a scalable bioengineering manufacturing, which has come out from our engineering members on campus. And so if you want a tour, just let me know. We can have great tours over on the edge of campus. The second is actually this genome technology. This is the CRISPR-Cas9 technology, which you've heard about in the news. It's really a bacterial system that can go in, recognize a particular part of the DNA that's bad. You cut the DNA, and then you put in a piece of DNA that tells the genome what actually you want it to look like, and then it will correct it without a scar. This is called CRISPR-Cas9 technology, and it corrects typographical errors. One of the world's experts is Matt Porteous, who's part of our center, and has been useful, again, here at Stanford to actually develop the technology for these pluripotent cells. So then if you have these cells that can make any cell in the body, you can actually use genome editing technology to correct it, and then use the tissue engineering technology to manufacture. That's the secret you need. And really the big breakthrough are these induced pluripotent cells, because you don't want to basically take axolotl limbs and then try to put them in people. Also, before there's a big issue about taking embryonic stem cells out of embryos, and that's unethical. And so Shinya Yamanaka, who won the Nobel Prize in 2012, developed a way where we can take adult cells and then using the Yamanaka factors, which we can talk about if you want to, and then you make those cells go back to look like those cells in the axolotl limb. And they're your cells, so you can they go back in time to the embryonic state, and they have the ability to make any, if you know how to coax them, to make any cell, any tissue in the body. Okay? So they can make unlimited quantities, so they can be scaled in our manufacturing facility. They're pluripotent, they can convert into other cell types, and you can actually genetically manipulate them outside of the body, so then you can correct genetic defects. So you don't have to do CRISPR embryos or CRISPR babies, which is unethical, and we can talk about that afterwards if you'd like. You can do it in a controlled and ethical way, then putting the cells back that are for you. Okay? So that's the idea. And so then we developed this stem cell biology where we can take some cells from your body, make them go back in time with the Yamanaka factor, and then correct them with CRISPR technology. So now you have a cell type that's you, that is just like the axolotl limb, which is pluripotent. They make any cell in your body. With our technology, you can then scale it in our facility, large clinical scale, and then coax it to go back into different tissues. In this case, for example, we can make skin, but as I'll show you in a little while, you can actually make essentially any tissue if you know what the right ingredients are to make the different tissues. And so this idea is what we're using for the Center for Definitive Curative Medicine, these induced pluripotent cells, or iPS cells, that provides a scale amount of manufacturing for making tissues that you can make for your own body. Okay, so as an example, this is a horrible disease where Stanford has been taking care of these unfortunate patients with recessive dystrophic epidermolysis bullosa, which is an incurable blistering skin disease. Basically, if you take your skin and then pull on it, there's one of 18 proteins that basically separates and keeps your skin on your body. So if one of these 18 proteins is missing, when you pull on your skin, it comes off. And this is one of these patients. This is one of these linkers, and one of them is actually collagen 7, this one right here, which basically is one of the big proteins that keeps your skin on your body. This is basically was first brought to Stanford by Gene Bauer, who's again here in the audience, and then it's been worked on as an act of love from our patients by Al Lane, who's here, by Paul Cavari, by a number of patients, all of our departments, and working on this, trying to help these patients, because if you've seen one of them, your heart breaks and you know you have to do whatever you can. And so actually it's through that love of the patients, and from Gene Bauer, Paul Cavari, Al Lane, Gene Tang, most of our department, they actually showed using a first technology where we can take a few cells off the body, put a retrovirus to correct it, that if you then had a keratinocyte, which is actually a keratinocyte stem cell, that was normal and genetically, if you took those, you can then put that on your body. It's one dose actually will cure the wound. Now 13 years and counting in some cases. That is the definition of definitive medicine. That is one and done. That is one dose is a cure. And that was done, but that took the preclinical basically diagnosis and shown that it could work in 1996 by Paul Cavari. After Gene Bauer had brought it, then Al Lane, Gene Tang, Pete Marinkovich, and others have been working on it through all this development for over 23 years. And now they're doing the phase 3 trial led by Gene Tang and Pete Marinkovich in our department to actually make this a therapy. But this is actually just a little set of skin. The question is, how can you scale this so then you can do a whole body analysis? And that's really where you go back to this iPS cell technology, where you can take these cells, correct the genetic defect, make this personalized iPS cell, and then coax it to go back to make skin. And that's basically what we've done. So now you can take these iPS cells using the coaxing factors, and I'll talk about more about these in a little bit. You can then coax them to make mature keratinocytes that are corrected, grow them up at large, massive quantities. In our GMP facility, we're using now technology which we borrowed from the bone marrow transplant unit, which is the other part of our center. And then you can purify these beautiful keratinocytes that are corrected and make tons of collagen 7, get rid of the bad cells that are not skin, they're actually not skin cells, and then graft them onto to show they work, and they make beautiful skin now with beautiful collagen 7 glue that keeps them on the body. So this is the idea, where you actually can coax them and make large scale. Now the question is, how can you then put them on the body? You put them on a little graft, but the idea then is to develop novel engineering technologies to then deliver them to our patients, which we're working on now. And the goal is to basically this is actually spray on tan, which many of our teenage daughters used right before the prom. But imagine if you have enough cells and the right idea to use a cell sprayer, which we're actually test driving now to actually spray these on our patients. Okay, so what about now? What can you do? What about, I just told you about skin. What about other tissues like blood, nerves, muscle for heart attacks, knee joints or ankle joints? And so what can you do? Well, with this IPS technology, you can then use, for example, we use two inducers, inducer 1 and 2. What these basically do is they basically take the cell and then fold the genome in a way that then can actually make skin. And there's basically about 24 of these inducers in the body. And it's kind of like having different ingredients, eggs and baking soda and flour, and you can make souffle or pancakes or omelets, depending on the order you add them and how much you add them and how long you bake them. And that's really the difference between this making skin, blood, nerves, fat, and muscle. And so a number of different investigators at Stanford who work on each of these different types of muscles have developed the recipe, if you will, like the Betty Crocker test kitchen, to basically induce iPS cells in these different, and so now we have a huge pipeline of not just skin, but other types of tissues, both wild type and corrected, for both genetic diseases and for tissue replacement for later on. And this is sort of the current list, not just keratinocytes, but composite grafts for wound healing, for diabetic wounds, cardiomyocytes for heart attacks from Joe Wu, thymic epithelium for a very unusual type of immunodeficiency disorder from the Weinberg lab and Katja Weinlach, bladder muscle for bladder incontinence from Bertha Chen's lab, and different kinds of neurons and glia cells for neurodegenerative disorders in Maurice Vernig's lab, Sergio Pasca, and the Palmer lab. And so really, the sky's the limit in terms of this cell-based technology. If you can figure out the recipe of these inducers and the order you add them, you can then make either genetically corrected or wild type tissues for genetic replacement. And so really, that's over in our clinic, the manufacturing facility over in California Avenue. And so with that, I just remind you what we've talked about. These three technologies that have sort of coalesced here at Stanford have allowed us to then finally address and make headway on these incurable monogenic disorders that affect people, common diseases with genetic components, and replacement tissues for wear and tear or accidents. And from the Maternal and Child Health Research Institute co-director, the idea is to then take kids that are born with these diseases and then try to take their stem cells out, correct them, and put them back, and then basically prevent them from a lifetime of just misery. That's really the goal that really drives what I do on a day-to-day basis. And so with that, just want to thank the people that have done the work, the Oral Lab, a number of different consortium and team science that we're doing across the country, the Center for Definitive Curative Medicine, and in particular, the support we've gotten from amazing colleagues of Stanford Dermatology, including Paul, Howard Chang, Pete Marankovich, Jean Tang, as well as the support in the past from Jean Bauer, Paul, and Al Lane. And so I'll stop and take any questions.
There are microphones here if anyone would like
Please.
to come on.
Tony, you showed the timeline of how long it took from the idea of replacing skin to when it's really
Right. happening.
Can you give me a feel for timeline for knee regeneration?
Great. Depends how long you run. That's the first thing. And I know how long you run, so it's going to be a long time. So the timeline from therapy to therapy, this 23 years, was because it's hard to get to the FDA. What they really want to do is every cell has to be defined, and that takes years and years and years. We need to come up with a way to accelerate approval in a way that's safe, but actually but allows these therapies to move faster. Because for a knee therapy, for example, it depends on what causes it. If it's just cartilage, then which I know for you is more than that, then that would be just one thing. But if it's different parts of the knee, then you have to work together. From the skin, we were making just for the EB patients. We know that if you correct just the keratinocyte layer, that's enough because the rest of it looks pretty good. For burns, they need both layers. For hair, you need both the epithelium and the underlying dermal papilla. And so those are, you have to, each of those is going to have an approval that's different. Yeah.
Tony, if a patient came to you with a good supply of cord blood when they were born, would that make any difference? Would that help you, not help you?
Good question. So yeah, so there's a big banking that goes on, and the question is that. So the answer is it might. And the reason is because we now take your adult cells, and we choose cells that basically, so between the cord blood and you've experienced some things in your life. The beach in Cabo, spring break in Florida, and so you've accumulated some mutations, some good, some bad. So cord blood is sort of pristine, and so the one thing that would be a very nice thing. The only problem is that your body's also is different, and so if you put those back, you might now have a rejection. So that's part of the issue we're dealing with now, is how to prevent the rejection, even though it's you. Yeah.
So he mentioned something that I thought was very salient. What is your path to get this to market to patients outside of clinical trials?
Yeah. So the path. So the question is, what's the path forward? And so the question is, so do you mean by regulatory path or?
A little bit of both. I mean,
Okay.
you obviously need outside people to help you.
Right.
Because you've got to take this and get it outside. So what are you doing to get it there?
Yeah. So the first thing. So the question is, what's the regulatory path for a cell-based therapy that's never been done? This is a first-in-human therapy. So the most important thing is that we make sure it's safe, by far. And so we've already had, for example, we have a very close relationship with the FDA, but it's sort of like my teenage daughter, asking my teenage daughter how she wants to be regulated on the weekend. So you want my credit card, you want my car, what time you want to come in. So there's this give and take with the FDA, because we're telling them what we think is safe, and then they're asking us questions about maybe you should make it safer. So it's this dance with the FDA first. Once it's safe, then we have to basically then find a market for it, and then from there, our approach really is this approach of rare going to common, because the quickest way for an academic facility to get a new therapy into market is to basically get FDA approval from orphan disease, breakthrough therapy, and now this regenerative medicine advanced therapy designation. That gives you an accelerated approval rate to get into clinic. And then from the platform technology, then you can then extend it out into different indications.
So what kind of timelines do you think that involves?
So the first therapy for our patients with recessive dystrophic, the question is what's the timeline? The milestone's looking like about 1.5 years to 2 years until. So it's not tomorrow, but it's actually from this therapy. It's actually pretty fast, and because of the FDA designations for regenerative medicine that they have now, it's actually quite fast. Jeff?
Tony, your work is just magnificent. Thank you for all of it. Recently, there was a report from Israel that they printed a heart out of stem cells. So it's not just laying a layer of keratinocytes on the skin.
Right.
There's some architecture involved. How is that done?
Yeah. So there's the 3D printers are pretty amazing. You can actually 3D print skin now with different layers. With computer-aided design, you can actually take a picture of somebody's hand and print the matrix. You can print a bladder. So if you print the matrix of where the cells sit on it, you can actually give a three-dimensional feel to the skin and the organ. So it's amazing. I mean, if you just walk across the street to the engineering quad and just spend 1 day in the product realization lab. My son was a mechanical engineer, so the PRL, the product realization lab, is like over there. It's like amazing, and so I think that's where a lot of this confluence of tissue engineering technology, which is out of my league, but which I have very wonderful colleagues, is where we're going. But that's actually, again, one of the beauties of Stanford is that it takes a village. You have your part of village, and then you work with these amazing people at Stanford, right? Cool. So the question is about other diseases, more common diseases like diabetes. So there is already a company where they take these pluripotent cells and they coax them using these inducers to make islets of the beta cells, islets for pancreas. And in animal models, they actually can regulate the blood glucose level already. So this is not science fiction. This is actually being done right now. They're moving it into sort of the clinical trials. So I think what I'm talking about here is not science fiction. It's today reality. Cell-based therapies where you have, again, definitive medicine, one dose, one and done is here. There are issues there in terms of how does the immune system deal with slight changes in the immune system. Those are all issues that need to be worked out. But the fact that you can actually manufacture beta cells, islet cells for pancreas, for diabetes, joints, bone, heart, and also brain for neurodegenerative disorders. These are all being done across the country, but especially here at Stanford. Yeah. That's a great question. So the question is, what about memory loss, Alzheimer's? So there's two issues there. One, we can manufacture those neurons that are, but the problem with memory is that it's a cumulative. The cells have to connect together, and that there's a long history of that connection. And so there's cell-based therapies now for Parkinson's disease, but which are being done right now. The problem is that are the connections correct, and is the history of that going to be like what you'd think of in terms of memory? And that's where a lot more biology needs to be done.
In the monogenic diseases that you're discussing, is there anything fundamentally limiting with your approach that impacts therapeutic intervention in utero for some of those conditions versus waiting till afterbirth?
Right, great question. So there's nothing that's limiting in theory, but I think so. Basically, technology in and of itself is not bad or good. It's how you use it, and I think, in my feeling, we need to be able to make sure that it's safe, effective, and delivery is actually safe before we move on. And I think the simplest way to do that is ex vivo first. And once we have some experience with that, then we can move on. And this is also the feeling of, so basically for all technology, especially for example, for using CRISPR technology for CRISPR baby analysis, that's completely unethical, and you need a large ethical board, which we have in our Center for Definitive and Curative Medicine. But there's nothing that can't be done. It's just that I think we need to do it stepwise. Great question.
Thank you, Tom. So Tony mentioned this, but I think it's worth underscoring in the context of his fantastic research. Really only about 6.5 years ago, this idea of turning back the clock and creating cells equivalent to the cells that existed when we first came into existence as a single cell fertilized egg in our moms, that has only been shown conceptually 6.5 years ago. So the work that Tony and is pushing forward in his own laboratory and as part of his directorship of the Center for Curative and Definitive Medicine, is truly at the frontier. It's unbelievably advanced, and it's pushing things forward. So Tony, I just want to thank you for that incredible lecture and for all the work you're doing. So before we break to go back and discuss some of the posters with our faculty, postdocs, and students, I just wanted to introduce some of the people who are going to be standing around the posters. There's a couple of them that might still be over there eating with the grad students, but mostly they'll rally again. We have Dr. Sumaira Asi. Can you wave, Sumaira? She is our Director of Procedural Dermatology. She's doing amazing work. We have Dr. Albert Chu. Albert is over there waving. Again, the faculty are by the food. They work hard. They need to eat. Dr. Justin Coe. Justin is over there. Fantastic. We have Dr. Tyler Holmig, who is here somewhere. Tyler, there he is, waving. Dr. Ann Chang. She is around somewhere. Dr. Galara Anari, who's waving there. So they're all kind of in the back in the corner. Dr. Jennifer Chen. Jen is there. Dr. Gina Kwan. Gina is sitting down. Oh, Gina was always a good student in medical school. Dr. Yun Kim is right there, another great student, thank you. Dr. Michael Kodadus is also sitting there. Wow, this is great. So these different faculty members are doing things related to skin cancer surgery, skin aging, eczema and skin rashes, irritating skin rashes, multidisciplinary lymphoma. And additionally, we have Dr. David Fiorentino, the very good-looking guy in the suit. Where is he? Yeah, see, he and I went to graduate school together. It's amazing, this guy. Dave Fiorentino, he is actually doing groundbreaking work in detecting cancer before you see them. Dr. Oro has some work here. Dr. Eleni Linos, one of our newest faculty over there, but was one of our trainees, went off, became famous, and now came back. We're really happy she's here. Dr. Betsy Bailey. Betsy was doing some incredible work in international and global health, our new director of global health. Dr. Howard Chang. Howard, where is he? He's getting applause, but I don't know where he is. He's somewhere around here. And Dr. Jean Tang. Jean is over there next to Eleni. Dr. Kavita Sarin. Kavita is right there, yes. You heard Joseph. Dr. Roberto Novoa. Rob is right there in the back. Dr. Susan Sweater. It's incredible. As I'm introducing each of these faculty members, they are world leaders not just in the science they're doing, but they're the people I send my relatives and friends to when they have a skin problem, and they're amazing doctors, every one of them. Dr. Carrie Lee, speaking of amazing doctors, where is Carrie? She's around here somewhere. Dr. Kristen Nord, speaking of amazing doctors, where's Kristen? There she is, right there, Kristen. And Dr. Marlianne Paul Rodriguez, another amazing doctor, right there next to Kristen. They were buddies from Columbia. Dr. Kevin Wong, Kevin right back there, eating, good to see that. Dr. Joyce Tang, our Director of Pediatric Dermatology. Joyce, where are you? I saw her earlier. Joyce, great. Thank you. Dr. Pete Marinkovich, a giant of a man scientifically and clinically. There he is back there. And also physically, actually. Pete is the only person taller than me in the department. And we already introduced Justin. So please take some time, talk to our faculty. They're here. They're excited to talk about their science. They're a little bit like the grandparents that want to show you the picture of their grandkids. If they get too carried away, just tell them to calm down. But we really want to close. Before you go off and see more and learn more, I want to thank each and every one of you for being here tonight, for all you do as part of this community, for all your support, your friendship, and your sustained engagement. Thank you. Thank you all for being here.